Hello everyone, I am Dr. Geetanjali Tomar, working as a women scientist at Savitribai Phule Pune University. Me and my team have been working over novel sources of mesenchymal stem cells since last 10 years. In our research, we came across a lot of tissues that yield stem cells and that can be used for designing cell-based therapeutic approaches for various degenerative disorders. While working over these stem cells, what we observed is as the age of the donor increases, the quality and the quantity of stem cells derived from those tissues, it significantly declines or tremendously it goes down. Moreover, it also becomes difficult to harvest these tissues from the individuals as they age. But while studying all these sources, we came across one such source which was the gingiva or the gums. And what we realized that it was not only easier to harvest gingiva from any age group of donor, but at the same time it was also yielding a large number of stem cells. They were able to retain their morphology in vitro, they had good growth parameters in vitro, and so basically with the age of the donor, the quality, neither the quality nor the quantity of stem cells was getting affected when we were deriving the stem cells from gingival tissues of such individuals. But keeping apart the growth parameters, although their differentiation potential or if I simplify it, their ability to form bone cells or adipose tissues, it tremendously declined. But interestingly, these cells, they very efficiently were able to form neurons. And therefore, in our study, we suggested that these cells can be used to address therapeutic designs for neurodegenerative disease. But it was not just about their differentiation potential. When we were working over this project, we were working under the conditions of lockdown because of the pandemic situation for the last 2 years, and we also thought of implementing our scientific research to contribute significantly to the clinical developments that were taking place during the COVID-19 pandemic. And therefore, we thought of studying the anti-inflammatory properties of these gingival tissue mesenchymal stem cells for lung inflammatory conditions. And for that, we developed a lung inflammatory animal model which was able to replicate the lung inflammatory conditions similar to the COVID-19 infection. And then we administered the mesenchymal stem cells in such animal models. And what we observed that although the gingival mesenchymal stem cells derived from older individuals were not significantly able to reduce the inflammatory parameters, but they were definitely contributing tremendously to the regenerative changes that the inflammation has caused in the lung tissues. And so finally we suggested that under conditions such as COVID-19, these stem cells can be used for designing autologous stem cell therapy for elderly individuals. Thankfully, finally our ideas and observations were recognized through our recent publication in Science Advances, and now we further aim at implementing these autologous stem cells for regenerative therapies, which in future means that as and when the individual they ages, their GUMS can yield stem cells and their own stem cells can come to their rescue for designing cell-based therapeutic approaches. Though we still have a very long way to go, but me and my team are continuously and extensively working to make stem cell therapy a choice or a clear option for common people and to bring stem cell therapy from bench to bedside.